Good afternoon, everyone. Welcome to Genotypic webinar series. And today we are going to have a webinar on decoding clonal evaluation and somatic mosaicism. It's a multiomic approach by BioSkryb Genomics. And I'm Dr. Karthik, Alliance Partner Manager, Genotypic. And Genotypic is the first genomic company in India, and we have a very good client base, and we have handled more than 3,000 types of samples, and we have analyzed more than 1 lakh of samples. And we are very happy to say that in India, we conduct the largest genomic conference, that is GIC, Genomic India Conference. And when we talk about genotypic products, so we have some own products, that is Commander software, GT GPUs, CPUs, GT Enrich, and GT Express. And when we look into GT Enrich primers, we have these set of panels ready in stock. And when you come into our partners, so we have multi-brand partners, and BioSkryb is an exclusive distribution. We have the exclusive distribution in India. And about the speaker, so we welcome Dr. Luca Maserati, and he's the channel partner manager EMEA. And Dr. Luca Maserati is the channel manager at BioSkryb Genomics. And before joining BioSkryb, he has extensive experience in single-cell genomics, and he has worked in QIAGEN, he has worked in 10x Genomics, and he also holds a PhD in life science, and he's really passionate on heterogeneity of these cells and disease evolution, and basically he's the man for single-cell genomics. So we welcome you, Luca, and the stage is set for you.
Sounds great. Thanks, Karthik, for the nice introduction, and good afternoon, everyone. Today, I'm going to introduce you BioSkryb Genomics and the new way of applying single-cell analysis. And this is the way that researchers are using BioSkryb Genomics. I'm going to present and provide you some examples on how those solutions, those reagents provided by BioSkryb are helping researchers in decoding the clonal evolution or the somatic mosaicism at single-cell level. So our mission clearly is to help scientists understand the immense cellular heterogeneity that exists and drive tissue biology and disease. So in this figure, it's showing that the mosaicism, it means like accumulation of mutations, increase in our cells as we age. And this is increasing not only in diseased tissue, but healthy tissues as well. So as the SMART consortium, which is the somatic mosaicism across human tissue, says, virtually every cell in the human body harbors a unique constellation of somatic mutations. While it is widely appreciated that RNA and epigenetic landscapes are dynamic, it's also becoming increasingly clear and exciting that our genome is not static. So, recognizing and decoding this genomic diversity, which is characterizing each one of us or each organism, is essential to develop the next generation of precision diagnostic and transformative therapies. So, single cell is really about clarity. And it is a requisite for interrogating tumor evolution or the heterogeneity and somatic mosaicism. On the left, what we see here is a bulk sequencing that gives us a noisy average data where, for example, somatic mutation, like the one here in green, get buried or confused with sequencing or amplification errors, artifacts. But on the right, the single cell sequencing really changed the game. It gives us a digital signal, a true heterozygous mutation that shows up clearly in 50% of the reads from an individual cell. This is the profile of an individual cells, which shows all the different, 50% of the reads on one allele, for example. So in this individual cell. So this is the level of precision that let us confidently survey the genome, even for rare events. And how we are able to survey the genome. So BioSkryb is the provider of a technology that is called primary template directed amplification, PTA. So we are mastering the way on how we can provide a great coverage and analysis in a uniform way of the whole genome. The way that PTA works is that we leverage an isothermal polymerase and high-fidelity TAC that binds short random primers, those short random primers that are hopping at multiple locations of the genome, which is our primary template. The polymerase starts the extension until encounters a terminator base, which terminates the extension. And this results basically in a collection of amplified fragments, which are in the average of 1,000 bases. And what is important to note here that our chemistry favors the primary template, favors longer template, like the genome DNA. So the polymerase, instead of reamplifying those short fragments, goes back to the primary template and starts other cycle of amplification, creating and generating other amplicons. So we amplify always from the same template, from the primary template, which is the genome. So this is a quasi-linear amplification in contrast to a very classic approach for whole genome amplification, which is multiple displacement amplification, MDA. And what is the advantage that PTA gives us? The main advantage is to obtain a comprehensive, unbiased, and precise whole genome sequencing, comparing to MDA, which is exponential amplification, and also because we are not reamplifying those fragments that we see here, the likelihood of amplifying an artifact, an error, is really reduced. And this helps us, or helps the researcher, to reduce also the sequencing depth when they are going to perform whole genome sequencing. And in terms of metrics, where are we? We are in coverage for in mapping of genome and genome recovery up to 97%, and with the highest sensitivity in detection of single nucleotide variants on the market. If we compare, as we did in our seminal paper published in PNAS in 2021, MDA versus PTA, we can see here a profile of the chromosome 1, where we have a lot of peaks. It means that there are a lot of areas of those chromosome that are over amplified, because this is an exponential amplification. But we see that this line goes often to 0. It means that those regions are not covered, are not amplifying, comparing to a more even and uniform amplification with PTA, where you have like really few peaks and almost no drops. Another important another important aspect of PTA versus MDA is that we are able to obtain a superior allelic balance. So ideally, you would want the allelic balance centered at 50%. So in the middle here, with half of the reads coming from each copy of the gene. For PTA, this peak occurs at 0.5. Conversely, for MDA, this is a wider peak representing certain alleles having a bias of amplification of one allele versus the other. So, and this is a comparison that we did it in 2021. There were also other papers, as I showed you, for the superior allelic balance that was that came up in 2022. Recently, there is a preprint provided by the SciLife Lab in Sweden at Karolinska, where they performed a comparative analysis of WGA kits for single-cell genome analysis. And they came out with the same results. So they wanted to compare PTA versus MDA. The key findings of this publication is that PTA provided a higher coverage and better uniformity with more genome completeness across single cell. They also showed about a lower allelic imbalance, so less dropout and even less amplification bias comparing to MDA. And in the detection of, they find out also improved detection of single nucleotide variants, copy number variation, so with a higher accuracy and consistency across replicates. Also, they find out that the kind of chimeric reads they were much lower in the PTA approach versus the MDA approach. So in general, this is just to show that is an independent evaluation by the SciLife Lab that confirms that PTA outperforms MDA for single-cell whole genome sequencing. What are the solutions that are provided by BioSkryb? Very easy. They're just two kits, and they are all reagents, not based on any specific instrumentation. All you can use any PCR and thermobath that you have already in the lab, and those are both manual workflow, but we also have an automated version for 384 reactions. So the two solutions are called ResolveDNA for whole genome amplification based on PTA, and then ResolveOme, which is the multiomic approach that allows you to detect whole genome and analyze full-length transcriptome from the same single cell. Both kits are compatible. If you want to save money on sequencing, you can do enrichment for exome or targeted panels, and we do have optional demonstrated protocols that are compatible, for example, with twist or IDT panels. The workflow from isolated cells to libraries is about 1 day, 8 hours. There are stopping points. You can stop in the middle of the workflow and you can resume the workflow the day after. But in general, this is something that has been optimized to be done in 1 day. We create and we provide reagents for library preparations. Usually those libraries are compatible with all Illumina short read sequencing. So we do create libraries compatible for Illumina, but you can convert the libraries to other platforms with the conversion kits. And we also offer a platform for data analysis, which is called the Base Jumper. Just to give you an idea of how ResolveDNA is used to detect genetic alteration in single cells in discovery in life science research. So in the first example, we have a model of acute myeloid leukemia, so all in hemato-oncology field. And in this paper, the researchers were able to identify single nucleotide variance difference between parental and resistant cell lines, including point mutation in the FLT3 genes, which is known to drive mutation. Or in solid tumor oncology. In this case, in primary breast cancer samples, we were able to identify losses in the chromosomal regions associated with common tumor suppressor. What I want to highlight here, that you are seeing here profiles, each profile is coming from a single cell. So what you have here on the top is the normal epithelial cells with normal ploidy, and you can see on the bottom there are other 3 cells that are showing a combination of loss of copy in various tumor suppressor genes. In neurology, also we have a lot of applications, and here in this very short picture coming from this paper, they demonstrated that the total mutation measured at the single cell level, and in this case they used nuclei, is increasing during the age, and this is happening in neurons. And also another type of application is in cell and gene therapy. When performing gene editing approach, like CRISPR-Cas9 approach, where you have a guide RNA to target a specific area to knock out a gene, then what you can do, if you have this powerful tool, which is ResolveDNA, that allows you to screen all chromosomes, whole genome, or the whole genome, you are able to detect not only at single cell level what is happening at the target region, but also all the off-target events like translocations, copy number variations that are happening on the whole genomes. And PGT is also another field of application when working with precious samples. Researchers are using this to characterize pathogenic variants. So those are some kind of examples, but I'm gonna give you and I'm gonna show you during the presentation few papers on how the researchers really used this technology. Before going into the applications, I just want to give you a highlight on the workflow for ResolveDNA. Everything starts with collection of single cell. We are a low throughput approach comparing to a high throughput approach based on microfluidic sequencing, for example, where you have like large number of cells that you can profile simultaneously and perform RNA sequencing. We are more focused on the genome, and we are focused on a small amount of cells. Those cells, they have to be enriched. And for doing the enrichment, then you will have to use a sorter to perform enrichment and isolation of single cell. So each single cell is going to be a reaction, and is going to be, the DNA is going to be amplified, and then one cell will be one library. So that's the switch where you are thinking. So we are thinking, as I said, a low throughput, but with a very high resolution. First step is the cell isolation. So you can do it through FACS, laser capture microdissection. If you're working with a cell culture, you can work. The important thing is that you can work with fresh frozen cell line. The cells must be viable. The DNA must be intact. It can also work with a microorganism, for example, or it can work with high molecular weight, low template DNA. We provide all the reagents in our kit for first step of alkaline cell lysis to disrupt the membrane, and the nuclear lysis to perform whole genome amplification. And after you have amplified your genome, then you can prepare the library and go and perform sequencing. And afterwards, data analysis. So this whole workflow has been optimized to happen in 1 day. And here I want to give you a first shot on how researchers in oncohematology are using ResolveDNA. So this is a preprint that was recently released in pediatric acute lymphoblastic leukemia from the Dan Landau at the New York Genome Center, and Chuck Gwad, who is our Chief Scientific Officer from Stanford, professor at Stanford University. In general, over 80% of children with acute lymphoblastic leukemia can be cured by treating them with multiple chemotherapeutic agents. But some of those cells can escape the treatment. So the question was, what are the rare clones that are escaping the treatment? They performed 3 approaches: a bulk error corrected sequence with on hotspot genes to detect, that was able to detect dominant mutation only. They performed analysis of single cell exome, so older multiple displacement amplification data, and a single cell whole genome sequencing using ResolveDNA. They used around, just to give you an idea, on the amount of cells, from each patients, they used around 100 single cell that were amplified around 10x for whole genome sequencing. And this helped them to detect 1,000 up to 2,000 single nucleotide variants per cells, including minor clones that later dominate or expanded under treatment. So they were able in this way to uncover rare clones that were previously undetected, linked to treatment resistance. And usually those type of research brings them to perform phylogenetic analysis to build trees to track the clonal evolution during therapy. So the key takeaway here is that PTA reveals hidden layers of mutational complexity and clonal evolution, enabling an early detection of resistance pathway and relapse. In measurable residual disease is also a kind of application, a field where we can apply, or researchers are applying the use of PTA. We know that a single clone can evolve in an invasive cells even after treatment. So this ultimately can lead to a development of a life-threatening tumors. So tracing single cells to predict which clones may persist or re-emerge after treatment is a way to characterize those, for example, CTC, circulating tumor cells. So in this case, MRD cells that have been enriched by flow cytometry can offer an excellent opportunity for genomic characterization of persister cells. And actually, this is what they are doing at the Dana-Farber Cancer Institute in Boston from the Gabriel lab. They used ResolveDNA to detect rare MRD tumor cells and subclonal genomic alteration in multiple myeloma patients that were not seen in bulk sequencing. The approach, this single cell approach, that brought this type of high resolution made possible to track the clonal evolution across the treatment, identifying persister tumor clones and therapy-associated mutation. And this is a recent abstract that was presented a few days ago at the American Society of Hematology. Other approach, which is in neuroscience. So in this paper, the authors, they wanted to assess whether chronic neuroinflammation in multiple sclerosis drives somatic mutation in single cell neurons from lesion and non-lesion brain regions. So the challenge actually was to accurately detect rare somatic single nucleotide variants in post-mitotic neurons, and this requires really ultra-precise single-cell whole genome amplification. And to solve the challenge, they adopted ResolveDNA, which enables this very uniform, comprehensive, and low error genome amplification. And the key insight of this publication was that neurons in multiple sclerosis lesions shows a 2.5 times higher mutation rate, suggesting that inflammation-driving mutagenesis may contribute to disease progression. Just to highlight that in this study, they were amplified and sequenced 73 neural nuclei using PTA. So just to give you the breadth and the idea of the amount of cells that are used for those type of approaches. It's not only a single cell approach, it can also be used, this approach, to amplify whole genome from a small amount of cells, like from 5 to 30, 50 cells. We call these like pseudo bulk or mini bulk, where we have, in this case, they researchers to characterize some specific mutations in targeted genes in patients with epilepsy. They used an electrode to puncture the brain of the patients, and then they take out the electrode, and on the tip of the electrode actually there are like few cells, so they wash off those few cells, and then they perform whole genome amplification using PTA. They were amplifying and enriching for the exome, but also the amplified genome was used for as a PCR input for a digital PCR approach to genotype some specific variants, in this case of the mTOR gene, that is linked to focal cortical dysplasia. So I was able in this case hopefully to show you how PTA is used in terms of flexibility, the type of input material that you can use, like single cells or nuclei in neuroscience, even a low number of cells, what we call it like this pseudo bulk, or even low input DNA. It works perfectly with all these type of inputs. And then what you can do after, once you have your whole genome amplified, you can use it for creating library and perform sequencing, or even for genotyping approach and specific multiplex PCR. Moving on the second part of the presentation, I'd like to introduce you the other solution, which is called ResolveOme, which is our multiomic solution that allows to detect or perform analysis on DNA and the RNA from the exact same single cell. And what that means, it means that we are able to have a better resolution. We can determine whatever genomic alterations can result in change in gene expression or even in the transcript structure. Or the other way around, we can focus on the gene expression first and then try to find out if those genes are influenced somehow by specific mutational profile of the genes of interest. So this is the really unique solution that allows you to couple genomic and transcriptomic information from the same cell. So studying single cell genotypes and with a phenotypic context, allows you to answer to some of those questions. So how the cell's genetic variants impact the gene expression, or what cell types have this mutation in these samples, because you are able to characterize the cells based on the transcriptomic profile. And how, for example, copy number or copy number variation and ploidy can impact gene expression. So just to give you an idea of how does the multiomic approach increase resolution. I'm showing here an example of an epithelial tumor. It can be like a breast tumor or prostate tumor. So we have the normal cells in pink, the pre-cancer cells, and also precancer in green, and precancer cells with an acquired mutation, and then cancer cells that become, during the invasive, when the tumor becomes invasive. So with an RNA sequencing approach, you are able to characterize the population of the cells based on the transcriptomic information, and you are able to characterize 3 groups of cells. And then you can infer the genotype from the expressed genes. But this is an in silico information that you are in vitro information that you are able to detect. If you are actually doing a multiomic approach where you have a unified genome and transcriptome analysis, then this helps you to have a higher resolution to better detect the cell population. So you are able to identify 4 groups of cells based on the information coming from the genome and the transcriptomic data. And the genotype is actually known. You don't have to do any analysis or in silico analysis to get the information of the whole genome sequencing. So ResolveDNA is the solution that helps you to perform this type of analysis. First step, same as ResolveDNA, you need to collect your cells. You need to enrich for the cells using a sorter. And after that, each cell is actually enriched and isolated in a well, then you can perform the process, the workflow using ResolveDNA, which is in 8 hours, has been optimized to be done in 8 hours. We always suggest maybe, especially at the beginning, you can split the workflow like in 1 day and a half. First step is a reverse transcription based on oligo-DT. So you can capture the mature RNA, messenger RNA. And then after the nuclear lysis, then you can perform the whole genome amplification based on PTA. So from the same individual cell in the same well, you have the genome and the transcriptome, so the cDNA and the genomic DNA. Then you can you can split the 2 portions, because during reverse transcription, our oligo-DT contains biotin, so all the cDNA is going to be labeled with biotin. Therefore, the separation can happen on streptavidin bits, and then you have your cDNA and the genomic DNA. For each of the portion, you can create a library. So at the end, from one cell, you will have 2 libraries, one for the RNA and one for the DNA, and perform sequencing afterwards and data analysis. One thing I want to highlight on the RNA performance of ResolveDNA is that the solution is designed to provide the full transcript coverage. So that allows you to detect also alternative transcript isoforms or gene fusions. Moving into the examples on how the ResolveDNA has actually been used in real world, here is an application of ResolveDNA on 190 nuclei from neurons obtained from temporal and frontal lobe. And in this case, the challenge was that some brain subtypes, the origin of some brain subtypes remained unknown. So they didn't know which was the evolution, the normal evolution of those cells, what was the origin, the anatomical origin of those cells. So they typed some of cells using ResolveDNA. In the nuclei, of course, you have to consider that there are only few mature messenger RNA, but still enough to perform cell type identification based on the transcriptomic profile. And they had also all the information of the somatic mosaicism, so the mosaic variants genotyping from the same cell. And this approach, which is a cell type resolved somatic mosaicism, helped them to uncover the origin of and the development of some cells in the human forebrain. A recent preprint from the SMATH consortium, where they were linking somatic mutation to cell identity and also their evolution, so their ancestors, with ResolveDNA. In this study, the SMATH consortium used the ResolveDNA to connect somatic mutation with cell identity and ancestry across three types of cells: skin, blood, and urine. So by sequencing DNA and RNA from the same single cell, they identified which cell types carried mutations and how exposure to UV or chemotherapy shaped them, shaped the mutational burden across the different tissues. They validated their findings using iPSC lines derived, so clonal cells coming from the same type of tissues, and using as well a strand-seq approach as orthogonal clonal techniques that confirmed the accuracy. So the key takeaway is that ResolveDNA sets new standards for linking cell type and lineage in somatic mosaicism, uncovering how environment and ancestry shape the human cells. I want to give you a case study, also a different approach. This is a collaboration that we did with the Duke University. BioSkryb is a US-based company. We are based in Durham. So we had this collaboration to support researchers on how to improve the prognosis in women with precancerous lesions. So how to identify molecular events that are driving transition from normal to ductal carcinoma to a more invasive carcinoma. What we did in this collaboration was the analysis of 12 unique patients. We collected about 80 cells per samples. All the patients had, let's say, different levels they were different level of carcinoma. They were like ductal carcinoma in situ, normal patients, or with invasive ductal carcinoma and triple negative breast cancer. All the patients were actually typed, genotyped, based on some specific markers like estrogen, progesterone, and HER2. I want to let you notice the sample number 4, which was typed as R positive, PR positive, and HER2 negative. So we'll come back on this. So 80 cells per samples. First approach, and they use the ResolveDNA. The first approach is, okay, let's have a shallow sequence, and with shallow sequencing, after whole genome amplification and sequencing, we are able to detect the copy number variation profile of each single cell that you see here at the site. And you see here the different chromosome on the top. So on the bottom part, you can notice the normal epithelial cells with a normal ploidy, while on the top you can already see the different cells. Those are the cancer cells that have different ploidy. And also we were able to identify few cells that had some discrete subchromosomal alterations. So this is just an approach with a shallow sequence. But if you want to go deeper and sequence at higher depth, then you are able to detect the somatic mutation. So the single nucleotide variants, perform single nucleotide variants analysis. In here, we have, for example, one gene, the ERBB2, which encodes for HER2, one of the most powerful oncogenes in breast cancer. And in two single cell out of 59 cells from the sample number 4, that if you remember, that was typed as HER2 negative, two of those cells, they are carrying a specific mutation, this S280F. This is an extracellular domain mutation classified likely pathogenic, which actually activates HER2. So maybe is not significant, having like only 2 cells out of 59 that are carrying this mutation, but can become clinically significant and a druggable target in a recurrent setting. And then here I want to show the power of connecting the mutational status with the gene expression, because it helps to reveal the cell state. This is a principal component analysis based on gene expressions. So the different colors you can see here, they are highlighting the different type of cells. So you have in golden the epithelial cells, in blue, for example, in dark blue, the stem cells, endothelial cells in black. So what you can notice here are all the diversity of the cell types that have been characterized based on the transcriptomic profile. Over here, we have epithelial cells that are actually clustering together, also because most of those cells are carrying a specific mutation in the gene PIK3CA, which is this mutation N345K, which is an oncogenic mutation for tumor epithelial cell. And here you can actually see the epithelial cells that are not carrying the mutation. Actually, those are the normal biopsies control cells that are lacking the mutation, and they profile slightly differently. One thing that I want to highlight is that in this group of cells, one stood out. This is a cell that shows a profile in terms of transcriptomic profile similar to a stem cells, but is carrying the mutation in the PK3CA gene. So this shows probably that this is a cells in transition, in the process of epithelial to mesenchymal transition. So that's the powerful on how to reveal cell states combining the two informations. And this is one last example. In this study, they used the ResolveDNA to profile, and this was a really high throughput approach because they profile this is from the New York Genome Center, more than 2,700 cells from normal aging esophagus, measuring both genome and transcriptome from each cells. And these allowed them to build detailed phylogenesis covering over 70 years of somatic mutation, and they identified clones carrying mutations in the TP53 and FAT1 genes, and repeatedly found 9q copy-neutral loss of heterozygosity events that tend to appear in basal epithelial cells. So combining DNA and RNA from the same cells was essential really to link this mutation to specific cell states and also to understand how certain clones gain a selective advantage during aging. In terms of data analysis, I mentioned that we have base jumper. So there are 3 versions of base jumpers. There is a cloud version, so you can take your FASTQ file after sequencing and upload your FASTQ file on the cloud. We also have all our pipelines for DNA and RNA analysis that are pre-uploaded, and you can download it on your local server. Otherwise, we also provide some custom bioinformatic tools as a service. ResolveDNA and ResolveDNA, they are both coming in 2 versions. One is the manual version, is in a 96 reaction format, but we also have the 384 format, which can be automated with Uno, the Tican Uno, in order to perform all the different steps of cell lysis and whole genome and transcriptome amplification and library prep. Is a semi-automated version to speed your work and to increase high throughput. ResolveDNA, we also provide the script for another liquid handler that is able to pick up really to handle micro volumes like the Integra Assist Plus. And just to terminate, we also have in US, it's working a lot, our service lab, where we have researchers that are sending us their cells, their samples, will do the sorting, will do the isolation, and then perform ResolveDNA or ResolveDNA workflow until library preparation, including with or without sequencing, and also with or without analysis, with all the different with all the different quality controls. So this is something that maybe in case you're able to ship sample to US, I know that could be quite challenging, but this is something that actually in US we provide as a service. And with this, I'd like to end my presentation. I want to thank you all for your attention.